Alright everybody, welcome back. Today we're going to be doing Lab 8, finally getting into viruses. Exciting stuff. The lecture will be heavily biased since my work focuses on that. But that's alright, hopefully you guys get a better understanding of it. So, first we'll just start by broadly defining viruses. First of all, a lot of people don't consider viruses to be living organisms or to be living at all, since they aren't able to reproduce on their own since they're obligate parasites. But some people do consider them live. For the purposes of this class, we will consider them to be biotic pathogens. So here we define them as particles that multiply only within living cells and are potentially pathogenic. Pathogenic means that they're going to be causing some symptoms in plants as a result of disease. They differ from other pathogens because they are non-cellular entities, so again that's why they're classified as just particles. And more than half of the known viruses cause disease. So that's the reason for that is viruses are parasites at the end of the day, so they're going to be messing with the with the metabolism of its host, taking its resources and such, and the host is going to have some response to that, and that's why what's going to cause a disease. Viruses though are very simple in that they have really all they need is replicase so that they can reproduce its DNA or RNA. That's the second component. And then the third is some type of protein to cover it, its co-protein. And that's really all a virus needs, just those three proteins. Sometimes most viruses have more than that, however, but really that's all it needs because some of those proteins that it encodes can have multiple functions. And here you can see what tobacco mosaic virus looks like, and this is what we'll be working with today. You've got this nucleic acid, which is RNA, kind of wound and in a sort of helix with the coproteins covering the outside of it. So here it's the nucleic acid is exposed, but there are other proteins, floating around in the cell that will bind to the nucleic acid. And what you get is this really long rod. Here's what it looks like underneath the scanning electron microscope. You can see they're just long flexuous rods, not flexuous, they're rigid rods. And that's just RNA inside of it covered up with proteins. I'm not sure what these balls over here are, but they're about 300 nanometers long, so they're very, very small. Now, plant viruses can cause a wide array of symptoms. Some of the more common ones, though, are up here. Most of the symptoms that plant viruses cause, the ones that we care about anyways, in either crops or ornamental plants, will affect its yield, if it's a crop, or its quality, also if it's either a crop or an ornament. So they don't typically outright kill its host because the virus is dependent upon the host. And you'll see they actually end up making the host kill itself, and I'll explain how the viruses do that in just a bit. But the typical symptoms that viruses cause are stunting, in which you get smaller versions of the plant, they don't grow as well. You can get mosaic as well because the virus messes with the photosynthesis of the plants. You can also have necrosis. The plant actually tries to kill the cells that are infected with the virus. So the plant actually ends up killing itself, and you get all these dead cells everywhere. And abnormal structures, and sometimes the virus will cause the plant to start to grow. And we think we know why viruses do that, but there's still some uncertainty in it. But what we do know is that abnormal growth is definitely a symptom of some viral infections. Now, viruses can be a little more difficult to diagnose because with most of the pathogens that we've worked before, well, all of them I guess, we've been able to see under a normal compound microscope, right? So if we can see the pathogen itself, a sign of the disease, then we know what's causing it. However, viruses, because they're so small, you need very specialized, very expensive equipment, and you need someone who's trained to use that equipment in order to see viruses. So that's what makes them very difficult to diagnose. So most of the diagnostics, initial diagnostics at least, for plants that may be infected with virus, are usually just looking at which host it has and which symptoms are present. So we know, for example, with rose rosette, with what I work with, if you start seeing certain symptoms in a particular type of rose that we know is susceptible to rose rosette, then we don't really have to, well, we should, but we don't really have to do much more diagnostics than that to confirm whether it's rose rosette. Some of the other methods that we can use to diagnose viruses are through immunological properties that it has. That's referring to antibodies, which will bind some particles that are specific to the virus. So that would be immunostrips or ELISA. We'll talk about that in later lectures. Electron microscopy, which is what we saw earlier. If you can see the virus itself, that's a pretty good sign of what's causing the disease. And then there's the biochemical properties as well. This is how the early viruses, or how viruses were diagnosed early on, by seeing what temperature could you inactivate the virus, just things of that nature. Now, plant viruses can be transmitted through many different ways. For example, there's vegetative propagation. Vegetative propagation is a means by which plants are reproduced or moved around that are contaminated with the virus. So, for example, certain species of plants can reproduce themselves by budding, and if that plant's infected, those buds that it produces can also contain virus and grow into a plant that's infected and produce even more virus. Or the seeds of a plant can also be infected with the virus, and just simply planting the seed will, start the infection process. There's also pollen. So some viruses will actually enter the pollen of plants, and depending on which species of plant you have, if you have a plant that's pollinated with that infected pollen, then you'll get seeds that are also infected. So there's that. There's also nematodes, fungi, and mites. So these two, at least nematodes and mites, feed on plants. So sometimes with their stylet, they can pick up viruses from an infected plant, and then when they go to feed on an uninfected plant, they'll spread the virus that way. Fungi as well, since they start to interact with plants when they break through the cell wall with their haustoria. Sometimes the virus can pass through from the fungus into the plant. Then there's insect vectors. So up here you can see an aphid that pierces the epidermis of the plant to get down to the phloem, and it can take in the virus. And some viruses actually can even reproduce inside its insect vector. A vector is just the name for whatever can transport the virus. So in this case, the aphid is the vector of BYDV, barley yellow dwarf virus. There's also mechanical transmission, which is what we're going to be working with today, in which simply taking sap from an infected plant like these tomatoes. If you look at these tomato plants that we have here, they have a lot of trichomes, or a lot of hairs on top of them. And if you break it off, just simply by rubbing your finger on it, you'll feel that there's sap on your fingers. So if a plant is infected, if one of these tomato plants is infected with a virus, simply touching the plant is enough to pick up virus on your fingertips in that sap. And if you touch another plant, another tomato plant, you'll also break the trichomes on that plant, and you'll mix that sap with that plant. So simply by touching stuff, and insects can do this too, such as aphids, when they start moving around on the surface of the leaves, they can transmit the plants that way. So the vector doesn't always have to take in the virus into its mouth and the rest of its body. It can just hang on to sap that's sticking to the insect, to its legs or something. And with these mechanically transmitted viruses, did this cut out? Probably the batteries. Let me know if it cuts out again. In the fields, just with typical cultural practices, these mechanically transmitted viruses can be moved around quite easily, such as when someone starts pruning plants without cleaning the utensils, they'll snip a branch off of an infected plant and then use the same shears on a healthy plant. Also, when transplanting, you can damage the roots, with cultivation as well, and with harvesting. So there's a lot of different ways that these mechanically transmitted viruses can be spread, and that's why it's important to have good farming practices that you guys don't need to know about for this class, but just know that they do exist and they're very important for this reason. So let's talk a little bit more about this mechanical transmission. As I mentioned, it's just somehow someone or something is able to pick up the sap from an infected plant, and this sap is where the virus lives, or rather where it's located in. And then the infected sap is introduced into another plant, but not just on the leaf surface, but it has to somehow penetrate the cells. So let's see. It has to come in through it has to contact the cytoplasm, which is the inside of the plant cell, or any cell. And the way we can do this is by putting minuscule abrasions, or just another word for it is tiny little cuts. And that's what we're going to be using in today's lab with a chemical, well, not a chemical, but something called carborundum, which are just very tiny pieces of carbon that are very jagged. So just by spraying it onto a plant or something and rubbing it, it will cause lots of minuscule abrasions through which viruses can enter. That's what we're going to be doing today. Now with infections, so after a virus enters the plant, why do we start getting all these symptoms of infection? Well, first the virus has to move throughout the plant so that it can spread and continue to use the resources of the plant to reproduce. So the viruses move throughout the plant, initially through the plasmodesmata, which, do you guys remember what a plasmodesmata is? Who remembers what it is? Anyone? Plasmodesmata. I see you raising your hand.
It's like how two cells are connected to each other.
Right, it's how all the cells in a plant are connected through tiny pores in the cell walls of plants. So, you know, the cytoplasm of the whole plant is technically all connected through the plasmodesmata. The virus can also move through the vascular tissue of the plant, you know, through the phloem. Do you guys remember what phloem conducts? Is it water or sugars? Sugars, right. So some viruses do go through the phloem, but the vast majority of them are through the xylem. Excuse me, the vast majority of them are through the phloem, and some move through the xylem. So with these with these local lesions, let's look at local lesions over here first. The virus is restricted to the point of inoculation, causing small necrotic spots on the leaves. So with local lesions, the plant is able to recognize which cells are infected with the virus, and it'll actually kill the cells so that the virus cannot spread. So, you know, ultimately this does hurt the plant, but if the plant only has to give up a few cells to get rid of a viral, you know, to prevent a virus infection, then ultimately the plant is better off doing that. However, sometimes the virus can outrun the plant, the cell death, and you start to get death all over the plant. You know, well, first, before the virus can move around, the movement of the virus throughout the entire plant is called a systemic infection, and by then it usually enters the vascular tissue of a plant, and that's how it gets to all the leaves and every single part of the plant, even in the roots. And this typically happens when the cell death response of the plant is not fast enough to deal with the virus. So this cell death response is called a hypersensitive response. It's a type of programmed cell death in which, again, the plant host will detect the virus inside of one of its cells, and will kill that cell and sometimes even the cells surrounding that area. However, sometimes the virus is able to outrun that response from the plant. And so what happens is, so if you look over here, we got papaya ring spot virus in this watermelon. And one thing I should mention is plant viruses are named after the first plant in which they're discovered in, after the first host in which they're discovered in. But that doesn't mean they're limited to just that host. So that's why you have papaya ring spot virus in this watermelon. And you can see, you start getting these ring spots. Ring spots are probably the best example to show of the virus outrunning the hypersensitive response from the plant. So the plant will start to kill the cells surrounding that infected cell. However, the virus will spread to the adjacent cells faster than that. So then the plant starts to kill the cells around those infected cells again and then again and again. And so what you end up with is these really nice ring spots. It looks like someone carved them, but it's actually just the virus. And then you also get them in papaya as well. So that's called the hypersensitive response. Let's look at some of the insect modes of transmission for these viruses. The most common, this is the most common method by which plant viruses move around. And these 6 up here are the most common insects associated with the transmission of viruses. So you've got aphids, whiteflies, mealybugs, and thrips. And then you've also got leafhoppers and grasshoppers, which can transmit viruses. There's also nematodes, fungi, and mice, as I mentioned, but those are not as common. Now, when we look at the types of transmissions that these insects can have, we need to take into account the fact that some viruses can replicate within the insect, and some insects can even, if they reproduce, while they're infected with the virus, their offspring can also contain that plant virus. However, sometimes, so what I was just referring to would be a persistent method of transmission, in which the insect will require a longer feeding time on the plant to intake that virus and then carry it internally until it reaches its salivary glands. So the salivary glands are where the virus is able to be transmitted into the plant that it feeds into to infect it. However, not all insects are like that. Some insects transmit viruses through what's called a non-persistent method, in which it acquires the virus by feeding on a diseased plant, and then it'll feed on another healthy plant and transmit the virus that way. However, only the virus really only sticks around in the mouth part of the insect, so it doesn't go inside of its body and start replicating. So insects that transmit viruses in a non-persistent manner are not such a big problem as the persistent ones. Alright, so now we're going to talk about tobacco mosaic virus, which is what we're going to work with today. Is there any questions then on the general characteristics of viruses? No? Okay. So TMV, tobacco mosaic virus, is the first to be discovered. It's one of the most stable viruses, meaning that it doesn't degrade when it's outside of its host. So viruses can survive outside of its host cells, but typically not for very long. Or it can not survive inside of living tissue. TMV has been found to live for they found a leaf, tomato leaf that was infected with TMV that was 50 years old and still had the ability to infect the plant. So it's very stable, very long-lived. It's got a wide host range, so again, it can infect plants that are not just tobacco. It's rod-shaped, as I mentioned, and the symptoms that it elicits once it starts an infection is mosaic, as the name suggests. You can also get mottling. Mosaic and mottling are similar, but slightly different, and I'll show you what that means in a bit. You can also get necrosis. That's when the plant can recognize the TMV particle and start to and elicit that hypersensitive response. That's how you get the necrosis. You also get stunting because the plant will start to lose some of that energy to the virus as it replicates. And then you can get leaf curling and leaf yellowing, which are again play a part in helping the virus to replicate. And it's also an RNA virus, so what you have here is a single-stranded RNA for its genome. And what do you guys think? What's important about single-stranded positive sense RNA? Think about it. Not just viruses in general, but what does it mean when I say positive sense DNA or RNA? You guys know what that means? Positive sense means it is the orientation in which the messenger RNA can encode something. So its genome, the genome of tobacco mosaic virus, is also a whole bunch of transcripts. So because it's a positive sense RNA virus, as soon as the particle enters the plant cell and it uncoats, it's ready to rock and roll. It can immediately begin to translate using the host's ribosomes and create viral products. So that's one advantage of having a positive sense RNA genome for viruses, and that's why a lot of plant viruses, the vast majority of them actually, are positive sense RNA. And then for TMV, they also infect Solanaceae. That's just the name of the family of tomatoes. So there's tomatoes, potatoes. What else is that? I think that one berry that's that one berry that was in Romeo and Juliet that they took to supposedly kill him. So that I forget the name of it. Blackberry, not blackberry, deathberry, something like that. Something poisonous. It's a poisonous berry. Oh, there it is, nightshade. Yeah, I should read the slide. And mandrakes as well, and eggplants. I didn't know eggplants were in that. All right, so how does the plant defend from viruses? Well, first things first, a plant will have preformed barriers to even prevent the virus from contacting its cells, right? So if the virus cannot get inside the cells, it cannot replicate. So that's why the plant will have preformed barriers like the cell wall. It'll have a waxy cuticle. So the cuticle is the outermost layer of the epidermis of a plant. And some plants have more cuticle than others. It's just a waxy layer. And when you look at like desert plants or something, you'll see that they look almost glossy underneath the sunlight. That's because it's all wax. Wax also helps with water loss and deflecting some of that sunlight. So that's why a lot of desert plants will have waxy cuticles. But it also serves a role in virus defense. And then bark as well. Bark is just very rough, dead cells for the most part, the outermost bark at least. So those are preformed barriers. But then there's also inducible defenses. Inducible, what does inducible mean? What do you guys think it means? Yes?
It can be like turned on and off.
Yeah, it can be turned on and off. And what do you guys think turns on these inducible defenses? A virus, right? When it recognizes, oh, there's a virus inside one of my cells, it's going to turn on some of these defenses. So it can produce toxic chemicals. Toxic chemicals, well first of all of these inducible defenses, they can be very different in what they do, but their purpose are all the same, which is to prevent the virus from replicating. Toxic chemicals can be toxic to the plant itself, such as reactive oxygen species, which are just like oxygen which has been reduced so that it easily reacts with other things inside the cell. You can also get pathogen-degrading enzymes. So some pathogens can transmit viruses to the plant, for example, fungi. So what a plant can do is it can start to secrete what's called chitinases. So you guys remember what chitin is, right? Chitin? It's found in the cell wall of fungi, and it's specific to fungi. So a chitinase, anything that ends with ACE, ASE, is an enzyme. So So chitinase is an enzyme that destroys chitin, so it destroys the cell wall of a fungus. And that's how the plant can, when it recognizes that there's a fungus on its surface or somewhere inside of it, it can release these pathogen-degrading enzymes. And then there's a programmed cell death, PCD. For the case of viruses, that's going to be called a hypersensitive response, HR. Alright, so now we're going to show you guys, we're going to get into the actual practical part of the lab. Are there any questions then on the lecture portion? No? Okay, so we're going to inoculate some plants with tobacco mosaic virus today. We're also going to finally start on your fungal identification project. For those of you guys that are online learners, you will be doing some other project in which you'll be answering lots of questions, hopefully to supplement what, or to replace what we're be doing here in the lab. What you guys here in the lab are going to be doing is you're going to be given 6 fungi that are unknown, and you will be doing a lot of work with them to figure out what they are. So this is where all your sterile technique training comes into play, because if you mess up and you contaminate some of your plates, you won't get the right results and you will lose points. So brush up on your sterile technique if you've forgotten what all that is. And then we're going to look at your tomato plants that you inoculated with agrobacterium a few weeks ago. Any questions then? No? Alright, let's get started.